Welcome back, friends. In this video tutorial we are going to talk about the different survival mechanisms of pathogenic bacteria, which is the Mycobacterium tuberculosis, which is causing serious health concerns in US and all throughout the world, because nowadays the disease caused by Mycobacterium tuberculosis, which is called TB, is very dangerous because it is very common in United States, and a TB infection is very much common in most of the cases. Now, the TB disease may occur certain cases, but the infection of TB is very much devastating, and if it left untreated, it can cause serious problems. So let's begin with that, and we will be talking about the different bacterial approach to cause a successful TB infection, TB disease also, and then we'll try to find out solution to prevent that. Okay, so let's begin. First, talk about the Mycobacterium tuberculosis pathogenesis and the overview of this pathogenesis. Now, first thing is that Mycobacterium tuberculosis is a bacteria. That bacteria used to reside in our alveoli. It used to reside in our lungs, in the alveoli regions of our lungs. They start to reside inside the cell, and they remain dormant throughout a long time. In fact, sometimes they can live throughout the life of the host individual itself. Maybe the host is carrying that particular bacteria throughout his life and he died, but still the disease is not onset. But sometimes the disease may emerge from that infective purpose. That is making it something unique about the infection of Mycobacterium tuberculosis, because it's not like that the infection is going on all the time. The infection is there, but it remains as asymptomatic. You can't see it, you can't visualize it. It may come out in certain times, and in those conditions it will cause the disease. So it begins with residing there inside the cell, and usually they reside there inside the phagocytic cells or macrophages. Now we know our immune systems are having cells to protect us against different bacterial cells or infections, and those cell is like one such cell is macrophage. So macrophages are phagocyting cells, so they are phagocytose all those bacteria like Mycobacterium or other type of bacteria. Now that's the job of those bacteria, those phagocytic cells, and kill those bacteria after phagocytosis. But what this Mycobacterium tuberculosis do, they target those immune system cells, and they take those immune system cell as their own home for residing there. So they are residing inside those killer cells, so that no other killer cell can recognize them. That's a very clever strategy taken up by bacteria like Mycobacterium tuberculosis. And normally what those macrophages do, they take up the cell, then they chop them up, digest that. But what Mycobacterium can do, they can reside there inside those cells without being degraded by the host cell machinery. That's a very important point, because they are having many important mediators to help them to spread the infection, to help them to reside inside the macrophage throughout longer period of time. And they can do this thing using several chemical mediators, several degrading enzymes like lipases, proteases, several degrading enzymes, and using several antioxidant and other materials. We'll be looking at each of these machineries to protect them against the host cell defenses in the future slides. So this is the basic overview. They stay there for longer period of time. When they find everything is optimum for their infectivity, everything is optimum for setting the disease, they just set the disease, and then it becomes difficult for the individual to cope up with it. Okay, so here comes the beginning of the structure of Mycobacterium tuberculosis. I must tell you, the cell wall structure of Mycobacterium tuberculosis is unique among the prokaryotes. You never find this structure in any other prokaryote cell, because in most of the prokaryotic cells you are going to find in the cell wall it is mostly made up with peptidoglycan. But in case of this Mycobacterium tuberculosis, you will find that most of the structure or most of the component part of the cell wall is made up with lipids. And very few part is made up with peptidoglycans. So almost 60% is made up with lipids. So that is telling us that it is made up with lipids. It's why lipids, right? So definitely presence of lipid is having very important significance in spreading the infection by Mycobacterium tuberculosis, right? So let's talk about in detail about the lipid structure. Now the cell wall structure of Mycobacterium, as I have told, over 60% of this cell wall is lipid made. And among the lipid structure, they are having three important components. One is mycolic acid, second thing is cord factor, and third thing is wax D. All of three are among lipids. Now first is mycolic acid. They are unique alpha branched lipids. As you know what are alpha branched and beta branched lipids, you must go on to my YouTube channel, you can find videos on that. So these mycolic acids are alpha branched lipids found on the surface of cell wall of Mycobacterium as well as Corynebacterium, which is another type of bacteria. Now they are kind of strong hydrophobic molecules in nature, right? And they form kind of lipid shell around the organisms. And what are the functions of having these mycolic acids or alpha branched lipids? This lipid shell kind of prevent the attack of mycobacteria by different cationic proteins or different enzymes like lysozyme and oxygen radicals, right? So this shell is helping them to prevent any kind of interaction or affection by proteins, lysozymes or oxygen free radicals. And also it usually protect the extracellular mycobacteria from complement disposition in the serum. It also help to block the opsonization process, right? So these are the functionality that bacteria gains due to having this mycolic acid layer. Now the second important thing is the cord factor. This is responsible for separating normally the serpentine cording on plates. That means the cord factor is toxic to mammalian cells, and it is also an inhibitor of polymorphonuclear neutrophil. So what we mean by that? Neutrophil, we know neutrophil is again a part of immune system which eventually recognizes the bacteria, uptake the bacteria, degrade the bacteria, and they can do all these things. Now this polymorphonuclear neutrophil means they are having different morphology in their nucleus, many different nucleus or many different lobe containing nucleus. So this polymorphonuclear neutrophil or PMNs are there, and these toxic mammalian cells this cord factor actually prevents those polymorphonuclear neutrophil migration inside our body, and as preventing the migration, it helps to prevent the uptake of these bacterial cells by those neutrophils. Okay, and the third important factor is wax D. In the cell envelope we can find this wax D. It's a component of Freud's complete adjuvant system, and this wax D is also a lipid, and it helps to this bacteria to escape from again the phagocytic cells. Now let's talk about the advantages of having the high concentration of lipid in the cell wall. What are the advantages? I must tell you, having cord factor, having mycolic acid in most of the cases are helping the bacteria Mycobacterium tuberculosis to escape most of the host defense machineries. That's a very important advantage. As well as what are the other advantages? One is that having all those mycolic acid layer and other things help this bacteria to not to be stained by any dyes. That's one of their advantages. They are impermeable to different stains and dyes that we usually take to stain other bacteria. Now the second thing is a resistance. It helps in the resistance against many antibiotics, because similar way the antibiotics cannot take entry through this hard lipid layer or kind of hydrophobic lipid layer. Again, the resistance to kill by any acidic or alkaline compounds. Similar aspects that alkaline or acidic compounds may not enter through this lipid layer. Resistance to osmotic lysis via complement deposition. We have already talked about the presence of cord factor. The presence of wax D helps them to block the attachment of different complement parts like C3A or C5A to be C3B or C5B or C2B to be attached to the surface of the bacterial cell. And finally, the resistance against the different lethal oxidants like oxidative damages, right? And also survival inside the macrophage, as they are having everything inside it. So having a lipid is definitely giving them the advantage to survive more than any other bacteria there. Now another important criteria, remember I've told you the first unique thing about the Mycobacterium tuberculosis is having more lipid content in their cell wall than any other prokaryote. Now the second unique thing about them is to have intracellular growth. Now this bacteria, this Mycobacterium tuberculosis, they can have intracellular growth. Remember I've already told you that they can literally grow and divide inside macrophage or host defense cell. Okay, so it's not just the bacteria is inside the cell destroying the cell coming out and then divide. It's not like that. They can literally grow and divide inside the bacterial cells. That is called the intracellular growth property of Mycobacterium tuberculosis. Okay, and this is an effective means of evading the humoral immune system. Why? Because as they're residing inside the cell, as they're residing inside the immune cells like macrophages, the host humoral response or the antibody mediated response won't be able to recognize the pathogen because it is inside the host cell. If it is outside the host cell and patrolling through the blood system, then only the antibody can recognize it, but in this case they are unable to recognize. So this is giving the bacteria another way of camouflaging inside the cell. Now what we know normally, we know that inside so normally if we look at here, if we look at this macrophage, and you can say that in this macrophage they are phagocyting the bacterial cells here, so they are taking up the bacterial cell inside the vesicle, inside the phagosome. After the bacteria enters inside the phagosome, it will immediately fuse with lysosome, produces phagolysosome, and lysosome supplies the important chemicals and enzymes. Those enzymes start to degrade the bacterial component, then those degraded bacterial components will be released outside. That's the basic way of how phagosome or phagocytes, macrophage usually function. But what happens in this case, to reside inside the phagosome or to reside inside the macrophage, they need to block this lysosome fusion with the endosome or with the phagosome, and they are doing this pretty brilliantly. How they do this exactly, we'll be talking about them in the future slides. Excuse me. Now what we can see here, the Mycobacterium tuberculosis appears to gain entry into the macrophages via cell surface molecules. The first line of interaction of the bacteria with the cell, it's required, it's important, and we know it is called the adhesion of the system. This adhesion is mediated by integrin family receptors, or CR1 or CR3. They are also called the complement receptors. These complement receptors like CR1, CR3, and the mannose receptor or mannose binding lectin or MBL receptors, they are also present there. Using this receptor, the bacteria start to adhere to the host cell like macrophage. Then they start to internalize inside the macrophage cell. Okay, so these are the process of engulfment. Now let's talk about the future stages. So what are the different stages that are being blocked by this Mycobacterium tuberculosis to survive intracellularly? The first important thing is that they need to block the each and a single different stage of the phagocytic response by the host macrophage, right? So in this case, the first thing you can see at this picture A, as you can see, they can block the binding of the phagosome with the lysosome, because you know that's the normal step, that's the first step of this process. So they can block the attachment or adhesion of lysosome with the phagosome. Second thing is that they can block the acidification of the phagosome. They can block the proton pumps that are present onto the surface of those vesicles where inside the bacteria present. They block these proton pumps, so that no acidification occurs inside the vesicle, and then the cell can't then the bacteria can survive pretty easily. Because if the environment become acidic, it will become hard for this Mycobacterium tuberculosis to survive inside those phagosome. To block it, it will block the proton pump. Third thing is that they can block it by producing reactive oxygen species, reactive oxidative species, because you know there are oxygen that can free radicals that can cause damages inside the bacteria. So they can block it by producing several enzymes like superoxide dismutase, like peroxidase, and several enzymes like that. The fourth round, they can also use, they can produce TACO, which is called tryptophan aspartate containing coat protein, and these are the proteins that are found onto the surface of the phagosome harboring the mycobacteria, so that it can modulate, it can bind with that cytoskeleton of the cell, the cytoskeleton of the phagocyte, so that this phagosome may not be able to move through the cytoskeleton to finally go and fuse with the lysosome. So it will again eventually block the lysosome phagosome fusion by modulating the cytoskeleton of the host phagocytous cell or host phagosome, host macrophage. And the final thing, the expression of virulence proteins, which are the example of virulence protein is PGRS, and we'll be looking at the detailed names for that and what are their functions. But several virulence proteins they can produce inside. They can damage the host cell machinery, they can damage the ability to produce enzymes to kill the bacteria and everything. So using all these important machineries, they can eventually survive inside the host cell. Okay. Now, inhibition of phagosome lysosome fusion. So let's talk about each of these different stages independently. First thing, that they can block the inhibition of phagosome lysosome fusion. And to block this phagosome lysosome fusion, they are having an important mediator, which is sulfatides. Sulfatides or sulfatide is an anionic trihalose glycolipid molecule produced by Mycobacterium tuberculosis, which is having a kind of anti-fusion effect. So it will block the fusion of lysosome and phagosome altogether. And also they can produce the defective activation of calcium dependent effector proteins. Example is calmodulin or calmodulin dependent protein kinase 2. So normally calmodulin or calcium dependent protein kinase 2, whatever, within the macrophages in the presence of mycobacteria contributes to the anti-fusion and the intracellular survival of the bacteria. That's another important fact here. Okay. Now the second process that they can block is the inhibition of phagosomal acidification. So they can not only block the fusion, but also they can block the acidification of the phagosome by blocking the proton transporter, because we know after the engulfment of the bacteria inside the phagosome, they start to induce the proton transporter so that the protons are pumped inside the phagosome to kill the bacteria. Now they can block exactly these proton transporters. So you can see here in this picture the proton pump is kind of blocked here. So within the Mycobacterium, the downregulation of vesicular proton ATPase pump in a lack of acidification phagosome is very important stage to survive for the bacteria. Okay, and in addition also they can produce ammonia to neutralize the proton environments or neutralize the acidic environment inside the phagosome, just like the process of Helicobacter pylori that we have talked. So usually this result in alkylating the intralysosomal components, and they can stay there after the neutralizing the environment with this ammonia. Okay, and we can also see that the presence of other channel proteins that they can block and try to produce other factors like some antioxidant, like some oxygen scavenging molecules and all these things. Third important stage to block is a maturation of phagosome, and this is very important because it's all about phagocytosis, is all about degrading the bacteria inside the phagosome and release it outside. So it can block the maturation of phagosomes. Now what we know that Mycobacterium tuberculosis residing within the host phagosomes modifies the maturation of the phagosomal components, and that enhances the intracellular survival of that bacteria in greater extent. Now for example, if we look at that, normally the expression of a protein called RAB5 on the phagosomes normally it helps in the maturation arrest of the phagosome in early endosomal stage, because we know at the very beginning the endosome, after taking up the bacteria, it's called the early endosome, then it starts to mature and fuse with lysosome called endolysosome, then the late endosome, and finally the degradation of the bacteria release outside. Now this maturation from early endosome to late endosome is mediated by several protein factors and several important chemical changes inside the phagosome. But the secretion of RAB5 protein by Mycobacterium tuberculosis eventually block this stage of the maturation from early endosome to late endosome so that bacteria can survive there. And finally, the recruitment and retention of TACO or tryptophan aspartate containing coat protein on the phagosome wall. So you know TACO, or another name of the protein coronin 1, is called an actin binding host protein. It's known to associated with the cholesterol. Now what we know here, the TACO is formed not only the surface of bacteria, it is formed onto the surface of the phagosome where the bacteria is in. As the TACO is present onto the surface of the endosome where the bacteria is inside, that endosome is kind of modifying this actin cytoskeleton, because we know for each and every movement of vesicles inside the cell it requires the actin cytoskeleton, because it's the movement of that vesicle onto the actin cytoskeleton that brings that particular vesicle from one place of the cell to another place of the cell, right? Now in this case, this taco is modifying the enzyme. The taco is modifying the actin cytoskeleton so that it is unable to walk through the actin cytoskeleton and finally fuse with the lysosome. So it's another way to block the fusion of lysosome with the phagosome, and also it's another way to block the maturation of the early endosome, right? And we also know that vitamin D3 and retinoic acid down regulates the production of taco. That's why vitamin D3 consumption is important if you are infected with Mycobacterium tuberculosis, because vitamin D3 will down regulate the activity of taco so that the bacteria can be degraded, bacteria can be killed because of the fusion of lysosome with the phagosome earlier. Okay, and and also the down regulation occurs through the modulation of gene via the genes called VDR and RXR response sequence present in the promoter region of the taco. Okay, now the protection against the oxidative radicals, it's called so protection against the oxidative radicals can be of two different types. One is production of oxidative stress proteins and the nitrate reductase proteins. Now oxidative stress proteins, for example superoxide dismutase, catalases, permeases, sorry, not permease, peroxidases, and also important different other factors. Now since phagocytic cells additionally produce oxygen radicals during the respiratory burst to kill the invading bacteria, it eventually supplies those enzymes to Mycobacterium tuberculosis in elaborated virulence factor, because it generally produces them to protect, and that's why they are supplying it. The enzymes found in Mycobacterium tuberculosis that combat oxygen radicals include AHPC and alkylating hydroperoxidase peroxide reductase that detoxifies the organic hydroxides or hydroperoxides, and also the enzymes or the protein products called SOD-A and SOD-C, the two species of superoxide dismutase enzyme that degrades the superoxides, because we know these are the scavenging molecules like superoxides. If they present there, the bacteria will die. So they produces SOD-A, SOD-C to go against those superoxides, and they also produces AHPC to go against hydroperoxidase. Okay, now the second scheme of our second system. So you can see here the production of different superoxides, catalase, and they are different blocking the DNA repair enzymes and everything. The second type of species are nitrate reductase, and they produces this nitrate reductase, and the examples of genes NARG encoded protein NARG, which can quench the reactive nitrogen species. And these enzymes also plays a key role in the growth of mycobacteria in the microaerophilic environment, when the environment is very much containing very much oxygen. This NARG protein helps the bacteria to survive in those condition of environments, especially during the later stage of infection, when the lung granuloma kind of formation start to occur, because in those conditions the individual is very much difficult, individual faces difficulty to take up oxygen for adequate oxygen for their supplement. In those conditions the bacteria get very few oxygen to survive. In those conditions this NARG proteins are helping them to survive in there. Now finally, the dormancy or persistence within the host macrophages. Now Mycobacterium tuberculosis has the ability to retain the dormant within the host throughout the time. That's another and probably the most important and unique thing about the Mycobacterium tuberculosis, that even it can reside in your body throughout your lifespan. You never know that Mycobacterium is there and disease is caused, because there is no diseased condition caused by the Mycobacterium. So we can simply call it a Mycobacterium infection, but not a Mycobacterium disease, right? So it may remain for years, at the same time retaining the potential to be activated. Now, the dormancy or latency of Mycobacterium tuberculosis allows the bacterium to escape the activated immune system. That's why they're doing it, because it helps them to escape from the activated immune system. Because when your immune system is activated, when the bacterium reside inside your body, your system is super active. You can find those bacteria, you can produce antibody against it, those antibody will go, it will it will help in the opsonization of the bacteria, phagocytic cell will go there, degrade them, kill them. But once residing there for several years and start to slowly grow and multiply in trillions of bacteria, then when every immune system response kind of suppressed in your body, they are not bothering about that infection anymore, they start to thrive, and all of a sudden, just like that, in very small amount of time, all of those trillion bacteria just come out and then cause the disease. Because in those conditions your immune system is so fooled, it definitely may not be able to interact and go against this bacterial infection. That's what the strategy taken up by Mycobacterium tuberculosis. Now, persistence of Mycobacterium tuberculosis is facilitated by several factors, and this is again under high active research, but several factors are associated with it, and one of them is isocitrate lyase, which is a glyoxylate shunt enzyme which is necessary in the lipid metabolism or fatty acid metabolism, and they are very good at fatty acid metabolism to manage inside the host cells and host tissues. Several genes were identified as being preferentially expressed when the mycobacterium resides inside the host granulomas or on the macrophages, and the examples are PE or PEPGRS gene families. The family encoding numerous repetitive glycine-rich proteins of different unknown functions. We still don't know the functions, but they produces glycine-rich proteins. That's what we get, glycine-rich proteins. And the mutation of those two genes, PEPGRS, produced mycobacterial strains that exhibited decreased persistence. So that is telling us that definitely these genes like PEPGRS are having a vital role in creating or in helping the dormancy of the bacteria in granulomas, right? So they are having a kind of direct role there. And also hypoxia was also observed to be a major factor in inducing the non-replicating persistence of tubercle bacteria. Okay, so hypoxia is obviously a major role playing agent in inducing the persistence of the bacteria. Once after onset of the infection, for longer period of time, hypoxia will set place, and that will help the bacteria, because bacteria can easily survive using those proteins. Remember NARG, right? So that's about the uniqueness of mycobacterium, and we have talked about the how mycobacterium modifies its own chemical synthesis and other kind of natures to finally go against immune systems. It's simply the way, it's simply the race between our immune system and mycobacteria, and it's mycobacteria that is going to win the race, because it is knowing our immune system very well and it is modifying itself according to that. That's kind of it, and I hope that's helpful.